So yes, this is on hormones, the way the hormones are regulated in your body. We'll go over the different types in this chapter. All right, in general, hormones integrate, they fine-tune these metabolic pathways that we've been talking about. They can be compartmentalized. They can go from one place of the body to a very far distant place. They can circulate. They can be really localized as well. So almost every process in us is regulated by at least one or more hormones. We've already talked about some of this, for example, insulin and glucagon. Remember, don't get confused between glucagon and glycogen. Glycogen is stored glucose. Glucagon is the antithesis, the Lex Luthor to insulin. So in mammals, they call the system the neuroendocrine system. What is the whole difference between the two parts, the neuronal system and the endocrine system, that they kind of merge together in an Orwellian way to make the neuroendocrine system? What's the big difference? Anyone know? And it's distance. Okay, it's the distance with which it travels. So neurons travel fraction of the micrometer, from the end of one neuron to the beginning of the next. Endocrines can travel. The endocrine system travels a large distance. Typically, they can be. Okay. Right. So that's why this is just a little figure from your book to show some of the different ways. Okay. Any questions so far? Like I said, especially this first part, a lot of it should be reviewed, I would think, from A and P. So how do hormones affect cells? How do they affect cells?
Bind to certain receptors.
They bind receptors, and so where can these receptors be? What's one of the locations for these receptors?
Outside of the plasma membrane.
Okay, extracellular. Where else can they be? So they can be extracellular. Where else can you have a receptor for something, for a hormone?
In the nucleus.
Nuclear. What's the third one? The third broad class. It's not in the nucleus, and it's not extracellular. Where's it going to be at, broadly? Cytosolic. Right. It's intracellular, but you can be more particular. Cytosolic, versus that way you can differentiate between nucleus versus the non-nucleus. Okay. There are at least 6 different intracellular consequences for this interaction. We've seen some of the ways that these can occur. So what are the different, and once again, intra means with N, don't get confused with inter. Inter, I-N-T-E-R, means it's between cells. Intra means it's within that cell. Okay. So what were the different 6, at least 6 that we can think of, ways that you could have a consequence? Just think about some of the ones that we've covered so far throughout this year. I think primarily this semester, but 6. 6. So I don't have polydactyly do the 6th finger. What's one way?
Upregulation, downregulation of the receptors.
Well, I mean, from the response. So from the hormone, whatever the neuroendocrine system signal, it binds to the receptor, and what are the different types of responses you can? Is that a real way of saying?
Phosphorylation.
Pardon?
Phosphorylation.
Okay, so yeah, receptor tyrosine kinase. You see where I'm getting at now? That's one of the ways. What are some of the other ways? Yes. And so that's with like the G protein coupled receptors, and so those are the cascades for the allosteric. Those can technically also be phosphorylation, but yes. Mm-hmm. What's another one?
INS.
The what?
The INS one.
INS? What was INS?
Insulin.
That's receptor tyrosine kinase. So that's 2 of the 6. What were some of the other ways? Think about with neurons. What's one of the hopefully obvious ones that happens in a neuron? How does a neuron signal get?
Depolarization.
Right. And so you have the ion channels. Okay. What's another one? So that where ion channels are membrane potentials. What's another really odd class? What's the one that can go both directions? There's only one that went both directions, both in and out into in. Remember what was it called? I thought that's what you're going to. You said INS, but not the instant slipper one. It starts with an I.
Oh, inositol.
No. What was it called? The one that starts with an I, where it's about binding things and it does that cytoskeleton changes as well as. Pardon? No, it'd be integral to the whole thing, the whole process of you living. Integrant system or the adhesions. Okay. What one have we not got? We got the one. What's the one that's nuclear? Steroids or steroid-like. And then the sixth one, which at least for me would always be the hardest one to come up with, and that's receptor guanylyl cyclases. Okay. So those are the 6 different cascades that we've seen. Different types of responses. From chapter 12, I think. So there are different ways that we can also categorize the signals. This is like the effect, the consequences, but you can also talk about their solubility. So now I just want you to look at or think about, whoops, let me make it smaller. You can talk about their solubility. So if you have those that are water soluble, sometimes they classify these as the amine hormones, since amino hormones typically are water soluble. I'm sure there are some exceptions. The amine hormones are the water soluble hormones. What you think about, do they pass through the membrane or are they going to typically bind extracellularly? And then what are some of the examples? Okay. So if you have a water soluble hormone, do you think it's going to be able to pass through the membrane or are they going to bind extracellularly?
Extracellular.
It's extracellularly. Why?
The inside portion of the phospholipid bilayer is hydrophobic.
It's hydrophobic lipid bilayer, so it's not going to be able to easily pass through unless it has some type of help. But typically they're going to bind to some type of extracellular response. So what's a good example of an amine or water-soluble hormone? What's a textbook example that we... Think about what was one of the examples we had that bound extracellularly to cause some type of intracellular change.
G protein receptors.
Okay, but what was something that bound to that? What was an example of its signal? Epinephrine, norepinephrine, or adrenaline, noradrenaline. Those are the amine ones. Those are the ones that derive directly from amino acids. What's another one that's a water soluble one? It's a peptide hormone. It did a receptor tyrosine kinase. Insulin, yep. Okay. But of course, you're going to bind have some type of binding on the outside. It's going to elicit some type of response on the inside. We've already talked about that. Sometimes it can also be things like the receptor the RGCs, receptor guanylyl cyclases. Just remember that one also has the possibility of having a cytosolic binding site. Do I get to answer a phone? It's been a long time since I've answered somebody's phone. Come on, that'd be one way to have a great week leaving. It has been a very long time. People learned. I used to answer phones all the time. Okay. All right. Oh, whoops, I didn't do it here. Whoops, did not mean to do that. Okay. What about those that are steroid or steroid-like hormones? Typically, where are they going to bind? First of all, can they usually, do they pass through membranes?
Yes.
Yes. Typically they can. I'm sure there is some exceptions. So therefore, their receptor will not be on the outside of the cell. They typically will usually have nuclear receptors. So what type of response do you typically see with these? Especially if you're going to do like a compare and contrast. At least for me, it's easier to explain those that have a nuclear receptor like a steroid. It goes through the membrane, it binds to a nuclear receptor. What's the effect that it typically has next? Expression. It's either going to upregulate or downregulate expression. It's some type of transcriptional control. Whereas if you compare that to those water-soluble, remember, think of insulin or adrenaline. You run it from the bear, but it had to bind on the outside and cause some type of change, or cause a cascade, which caused this, which caused that, which go on and on and on and on, before it affected. It wasn't like more of a direct effect transcriptional. There's this little cartoon that shows that from your book, where REC is for receptor. So we can see the one on the right, the little, kind of like a jigsaw puzzle, but it's the steroid tetracyclic creams of the steroid that passes through, binds to some type of nuclear receptor, which then alters transcription. Whereas for something like insulin, you have to bind externally, causes secondary messengers to occur, which those will either alter pre-existing enzymes, or then they can go on to affect transcriptional control. So sometimes people have talked about, like the relative speed with which some of these alter. And it's kind of a trick question. You'd almost have to give a specific example. Because if you're talking about transcriptional control, then maybe steroid would go faster. Whereas if you're talking about altering the activity of an existing enzyme, then it may not, right? Because the steroid one is going to be slower in that sense, because they have to synthesize or downregulate the expression of that enzyme. That's why. Okay, then we can even further classify according to the method of delivery. I used to ask people to this, but found out that most people have forgotten from AMP the difference between endocrine, paracrine, and autocrine. Autocrine is the easiest because remember, auto means self, and so it means it affects the receptors on the same cell. The paracrine means it's within the neighboring region that it affects its neighbors. Endocrine it can affect long distances. Or you can classify them according to their structure or their origin, sort of like what we've been doing so far with the amines, things like histamine, serotonin, things like that, versus peptides, which would be glucagon, insulin, somatostatin. Okay. Oh, that's right. I did it differently. Okay, than what I thought I did. Are there any questions so far? Like I said, a lot of this first one is definitions. So it's one of my least favorite. I actually like this chapter a lot. It's just that this first part, you just have to hopefully refresh your mind over stuff you should already know. Okay. So of the classification, or this type of classification system, there's scientists, they like to classify lots of things. You can see the different types here. We have peptides, catecholamines, icosanoid steroids. Vitamin D, retinoid, and retinoid ones are kind of little asterisks, just because, well, you'll find out in a moment. Thyroid, nitric oxide. Okay. And we're going to break them down piece by piece. So, first up, we have the peptides, things like insulin and glucagon. So, how are they made? Do you remember? We talked about insulin some already. How is insulin first translated? When it's made, it's obviously it's a peptide. But what kind of form is it made in? Do you remember? Like, how do you control insulin for a healthy person?
The amount of ATP.
Well, I mean, what does it look like when it's first coming off the ribosome? What does it look like? What form does it take? Is it what? Well, it's got a primary structure, and what is its primary structure composed of? Does anyone remember what it's called?
Trying to say it's like globular or something like that?
No, I mean, it's called a pre-proinsulin. It's made in a precursor form that's got to be proteolytically cut up, right? That's how we actually control it. So these usually have prohormones. Insulin actually has a pre-pro hormone, but it's made this really big, big is relatively speaking. But it's made in a larger size and it gets cut down to its active form. Okay, so it's cleaved. When we already talked about the fact that it's going to have secondary messengers and bind to the outside of the plasma membrane. So this is the example of insulin. I do want you to know the details on how insulin is made, just because there are so many health issues involved with insulin. Okay. You should already know, kind of speeding along, what organ makes insulin, and it's the pancreas. The pancreas makes other hormones as well. What's another hormone the pancreas makes? There are two other biggies. My word, not the textbook word. Insulin. What's the other one that it makes? Most people can remember the second one. Glucagon. What's the third? The one that I call the redheaded stepchild of the three pancreatic peptide. The one that no one can remember. The cousin Oliver of the Brady Bunch. That's before your time. I don't know if you guys watch Brady Bunch. I can't think of anything from your generation. Okay. But your parents would know exactly what I'm talking about. So, maybe grandparents, I don't know. So, it's the metastatin. Okay. Then people, so we'll talk a little bit about it. We'll talk a little bit about it. Even in this chapter, we kind of just gloss over it. Okay, so typically when it's first made for insulin, it's made in what's called the pre-pro form. And it doesn't make a lot of sense, the fact that they call them the A, B, and the C peptides. And in fact, especially since it doesn't go in order, I don't know why they kept those names. I can hypothesize. Because, normally we go from the amino terminus to the C terminus. But if so, it goes BCA. Okay, but there's also a signal sequence that helps target it for its localization as well. Okay, so it's first made in the pre-pro insulin form. The signal sequence is what puts it into the secretory vesicles. Unless you're in Canada or the British, it's vesicles. But the secretory vesicles. And proinsulin is the way it's stored until ever it needs to be used. And it is stored at such high concentrations that it's almost crystalline. You can actually store proinsulin in really, really high concentrations within that vesicle. Now, whenever your blood sugar, you should know whenever insulin gets released, so whenever your blood sugar increases, it's going to signal for the C-peptide portion to be removed. So that's why I hypothesize this, right? It's called the A versus the B. Because whenever it cuts it off, you can see it cuts off here and here, it's literally only held together by disulfide bonds, which makes it unique in that sense. And then it's going to be active. I thought I had another picture coming up, but not the picture I thought. It's in these vesicles for whenever it gets released. Okay, be released. Exocytosed. Insulin is not the only one, and there are certain genes. Here, let me just show you. A single gene can make a lot of different hormones. So this is the POMC gene. I would not ask you what it stands for, because I'd have to look it up. But you see, POMC gene is huge. Of course, you should already know by now that the direct transcript of it already is smaller than the original gene. And then if you look here, there are proteases in different, for those of you who've had entomology and neurochemistry, you already know a little bit about this too, that can cause alternate splicing sites. So there's also structural elements as well that can cause it to cut in different locations. And from that one gene, you can get a whole host of different hormones. So once again, I would not ask you, okay, beta MSH, comes from the POMC gene, and if it's not cleaved, it makes lipotropin or anything like that. Okay, so please. I would have to give you this figure, or I could ask you just qualitatively something about this, but you don't need to know. But yeah, these proteases can know. Sometimes they're known due to locations of the positive charges. Sometimes, not with this one, but sometimes they're also known where splicing can occur due to structural integrity of the original gene as well. Okay, are there any questions on Okay, so as I mentioned, go back to hormones, some of these other peptide hormones, insulin and some of these other peptide hormones. The prohormonal form typically are really high concentrations. So then it goes through exocytosis and it gets released into the local environment there. But the way it gets picked up the way the insulin gets picked up to be shipped off to the rest of the body is the fact that the capillaries that are right by those endocrine glands are fenestrated, which is a fancy word for what? It's a scientific term.
Holy.
What? Holy. Yes. Holy. Like cheese. So that's why they're permeable. The peptides then just flow into the capillaries and then can be taken wherever it's needed. Right. The next section, if you're going on the classification system, because I think I accidentally uncovered it before, are the catecholamines. An example is epinephrine. So catecholamine. So here, let me write it out, just because I don't want to give away the answer before we. This is a good review. So catechol, by the way, did you guys study what catechol was in organic chemistry? It's an organic chemistry term. It's a compound. But catecholamines, an example for it is epinephrine. Epinephrine. And what was its precursor? Where does it come from? Tyrosine. Mm-hmm. So the catecholamines comes from tyrosine. Usually, epinephrine's made, or usually catecholamines. So just think of epinephrine. What tissue makes epinephrine? It's adrenal glands. Always think about what's really called adrenaline as well. So it's adrenal glands. And they're stored and released very similar to the peptide hormones. Okay. So they are exocytosed, stored in vesicle formation, and they're exocytosed. And I didn't check the most recent edition, I'm sorry, see if it's still table 23.6. That number may have changed. If you have the, was it 7th edition now? So we've covered the peptides, the catecholamines. The next group are the eicosanoids. Okay, so the eicosanoids. Does anyone remember what compound is the precursor for the eicosanoids? You have to go back to earlier this semester. It's arachidonic acid, arachidonate. Mm-hmm. So one example is the prostaglandins is PGE1. I would expect you to know how to draw it if I give you the information. Like this one, I think it's 5, 9, 12, and 15, but I may be wrong. But I also have to give you the location to remember what kind of double bonds are they in arachidonate? Are they cis or are they trans? Cis. They're always going to be cis. So those are your prostaglandins, thromboxanes, leukotrienes. The big difference between them and all the ones we've discussed so far is that they are not made in advance. We don't make them and store them in vesicles just waiting for release. Okay? And these are paracrine hormones, which paracrine means there's autocrine, endocrine, and paracrine. Autocrine means it acts on the same cell it came from. So what's paracrine? In the local environment, the neighboring cells, versus endocrine would be R. Then we have the steroids, which I don't know why I messed up the boxes. The steroids, example of steroid hormone. What's an example of steroid hormone? Testosterone. And it's synthesized from Where do you get the steroid hormones? Pardon? Cholesterol. Testosterone is all your androgens, estrogens. I'm not going to go into a lot of more detail about that right now. We can then look at the vitamin D ones. This, I would never ask you to name it, spell it. Yeah.
So when people take test boosters, would that increase their cholesterol levels going backwards?
I don't know. First of all, if you're just doing it for a short period of time, I doubt, unless you're really susceptible to that, I doubt that you'd have to worry about it. If you have a family history of it, you probably shouldn't. If anything from Lavoisier is, the shot lady's principle, to where if you remove it from one section, you're going to increase it. But I don't know where those come from. If they come just directly from the cholesterol, or if they're all the way back from the methylonic acid, methylonate from before. Where does vitamin D come from? What's its precursor? Pardon? The sun. Well, no. It activates it. By the way, this was just a form of vitamin D. I wouldn't ask you, like I said, I wouldn't ask you, okay, draw 125 dihydroxycholecalciferol. Where's it coming from? What's its precursor?
Steroid.
It is a steroid base, so therefore it would come from cholesterol as well. And I kind of gave this away. I think the very last class I was talking to some people afterwards about retinoic acid. So the retinoid-based hormones, an example of retinoic acid, what's its precursor? You remember? It's really surprising you know this from. There are 3 based. So we're like,
Vitamin C.
it's not vitamin C because vitamin C is. It's vitamin A. Retinoic acid, retinoate, is vitamin A. Vitamin C is ascorbic acid or ascorbate. And then vitamin E are the tocopherols. Okay, so yes, it's vitamin A. And then the last 2, thyroid, comes from tyrosine via the thyroglobulin. And nitric oxide doesn't really fall into the, I don't like the way that they have this one. Nitric oxide is nitric oxide. And it comes from nitric oxide synthase via arginine. So it's a little bit different. Okay. Are there any questions so far? Are you doing good?
For this whole table, do we need to know all of that? Or
Yes. I'm not, like I said, I wouldn't, vitamin D, you can put down vitamin D. Retinoic acid is vitamin A. But I wouldn't ask you triiodothyronine, some of those. I would expect adrenaline and noradrenaline and insulin, glucagon, somatostatin, that kind of stuff as we go along. So,
along with the mode of action as well.
right, because those are the ones that we've actually already covered all of this. So, because if you know that steroid hormone, which also includes vitamin D, and then if you think way back to the lipids last semester, remember there's 4 vitamins that are fat-based, they're lipid-based. Do you remember what they are? DEAK, D-E-A-K. And so that one, since you know that one's vitamin A, then you know that that has to be nuclear receptor. Because those are a fat base, those are all lipids. So if you just start to learn about logic, rather than just memorize this, if you learn the logic, these are peptides. And then, that was a little special. These are peptides, so they're going to be water-based. And they're going to have a positive membrane. These are the steroids, so they're going to have nuclear. Okay. Nitric oxide is the outlier because it's the one that works with the cyclic GMP. That's the cytosolic form. Okay. Let me make this smaller. So the gland that oversees all this is the hypothalamus. So again, like I said, most of this should be review. So if we're going to look at the anatomy of this real quick. We're going to focus on the hypothalamus. Here we go. I would expect you to know roughly where it's at, meaning, like the part of the, like if you could just draw this and know that the hypothalamus is right there in the center. Oh, I don't know what happened there. Sorry. Oh, whoops. Here we go. The hypothalamus is like this region right here. Then you're good. I'm not going to ask you to actually draw out the hypothalamus, but these are the major, glands that are important to this. So we have the hypothalamus, and down below is the pituitary. The thyroid is right there in your throat. Of course, the adrenal glands are going to be on top of the kidneys. They kind of look like little dunce hats on top of the kidneys. We have the pancreas, which is located behind the stomach. And then we have the sex organs as well. Okay. Oh, and then adipose tissue, which could be in various places. They're just showing it there in the love handles region. So if we blow this part up, please don't blow it up, but we can then look at it and we can see that the hypothalamus is a relatively small region just above the, or just anterior, just above the pituitary gland. And then we can divide the pituitary gland up once again to the posterior and the anterior. Anterior is in the front, posterior is in the back. And then we can actually look at it in further details. Let me move this up so we can see it. And this is the level with which I would want you to know things. I will not ask you, and you'll see it in the next one, which are posterior versus anterior hormones. I will not ask you that. Which of the tropins come from which direction? Okay. So this next slide, other than I want you to know the big scheme of things here. So typically you have the input, and we can divide it up, like I said, to the anterior to posterior. I'm not going to ask you oxytocin comes from anterior versus posterior. However, I would expect you to know what oxytocin does. Oxytocin is involved in smooth muscle, relaxation. So that kind of thing. That's the extent of which it. So typically we have neuroendocrine system. You have your first target, secondary target, and the ultimate targets. Some of these don't have as many steps along the way. And then what you can see here is the fact that they can be, some of them are in a precursor form, the tropin form, versus the fact that they get modified throughout the entire process. Okay. But I just want you to know the ultimate targets in particular. Know that it's the hypothalamus that regulates everything. It signals down to the pituitary gland. From the pituitary gland, I would expect you to know, especially those that are the androgens, the sex hormones. We'll cover actually gonadotropin just a little bit. Oxytocin, vasopressin, and then the ones that we've already covered before. I said gonadotropin, it's somatostatin. I'm sorry. Are there any questions so far?
Is thyroid-tropin also the same thing as TSH?
Thyrotropin, the same thing as, well, the tropin, let's see, where's this one at? Thyrotropin. Whoa. Sorry. Thyrotropin, the same thing as TSH. TSH is the stimulating hormone. And so I'd have to double check that one myself.
I don't see that there.
No, and I'm wondering if I'll double check, see if that's the same thing or if it's going to be a precursor for something else. So the way we did with the I'll just double check that one to see if it really is the same thing as thyrotropin or if it's one of the alternate forms, like a precursor form. Or technically, thyrotropin would be the precursor form of TSH. Okay. Here we go. Oh. So I always like to point this one out. Actually, I think I've pointed this out. Usually when we learn about peptide primary structure in the beginning, just because some of these are such small differences in them. So this is the difference between oxytocin and vasopressin. They are both very, very small hormones, short peptide hormones, but they only have two differences between the two of them. So one's isoleucine to phenylalanine, and the other one's leucine to arginine. This first one isn't considered that huge of a difference because, this is a branched chain hydrophobic amino acid. That's an aromatic hydrophobic amino acid, but this is the big difference. That's a positive charge. And of course, this one is another branched chain hydrophobic amino acid, but they have very different effects. Then we have the tropin, which as mentioned before, those are just really long polypeptides that many times will signal onto additional things or get modified in the process. I always want you just to appreciate this. Okay, do not memorize these numbers, please. But to see that we have an initial signaling event, and what it does is going to activate the hypothalamus to release this hormone. This one that they're doing has to do with cortisol. So it's first corticotropin releasing hormone. It's only in the nanograms. This goes on down to the anterior pituitary, which it releases ACTH. It's now micrograms, which goes down to adrenal gland. Adrenal gland releases cortisol in the milligrams. And so we went from something that was a single event to all the way down to in the milligram kind of concentration. So you can see where it's a cascade, similar to what we did with adrenaline during chapter 12. Of course, then the cortisol is what's going to go on to affect the tissues that it's going to affect. Cortisol builds up, it's going to go back here and inhibit both the hypothalamus and the interior pituitary. Okay. Then I always try to just really quickly wanted to show, and you can look at these. I did make sure, I know this one for sure goes back to the journal article. I'm sure surely this one is. It's called the hypothalamic pituitary adrenal axis, this HPA axis. It's a relatively quote unquote new issue, but some of the indicators. Some of them are trying to say that you can look at this at infancy or possibly even in utero to predict adult diseases. And then finally, then there's a possible genetic defect that was found within the last few years. And so I'll put in some articles there, but you can see that it's really interesting at least to see if they can possibly predict